This is Boston University's Neurophotonics podcast. My name is Sung An, a second-year PhD candidate in biomedical engineering, and I am the producer and editor of the podcast. This episode, our marvelous hosts are joined by Dr. Ryan Raut, as he discusses his journey from an interest in psychology to studying brain state dynamics and arousal. He also shares powerful insights into the nature of advice and exciting future directions in neurophotonics. So, thank you for tuning in, and please enjoy the episode.
Hello, everyone. Welcome to another episode of the Neurophotonics podcast. My name is Dora Balog. I'm a third-year PhD candidate in biomedical engineering, and I'm here today with my co-host,
Sudan Duwari. I am also a third-year biomedical PhD candidate.
We're here today with Dr. Ryan Raut, a Shanahan Fellow at the Allen Institute and the University of Washington. Ryan is interested in the general principles of how brains work across different spatial temporal scales and species. His approach comes from a multidisciplinary standpoint at the intersection of neuroscience, dynamical system theory, and scientific machine learning. Welcome, Ryan. It's good to have you here. How do you like BU so far?
Thank you so much for having me. First of all, I'm really excited about this whole visit and this discussion we're about to have. And I think I've only been here less than a day so far, but I'm really enjoying the people, the community here, and especially the sunshine coming from Seattle around this time of year. So,
we had some pretty bad rain the other day, so you're very lucky.
Okay, welcome from my side as well. Let's get started with the education. So how did your academic journey look like?
Yeah, okay. So my academic journey, let's see, I guess I'll start around college time, which was when I started kind of narrowing down in interests and such. And I did my undergrad at the University of Wisconsin-Madison. So I grew up in Wisconsin, and that was a natural choice. And that was around the time when I started to become really interested in psychology. So that's actually, I came into neuroscience via the psychology route. And I think at the time it was mostly just, I think all of us have that kind of intuitive interest in psychology, right? And understanding other people and kind of realizing that maybe our behavior and the patterns of thought and such are not totally random, not totally up to our free will, as it were, but that there's some rules and regularity to that. So I shared that interest. And then I think at a certain point, I took a class maybe my sophomore year, later in my freshman year, actually, that was in the space of cognitive neuroscience. And that was when I first kind of became familiar with this modern ability we have to actually study the human brain in vivo and to take pictures of your brain and such. And I just thought, what a time to be alive, right? Like that we live in a time when we can do this. So actually, until that point, I never liked science. In fact, I would say I strongly dislike science. I even took a biology. Everyone has to, right? I took a required introductory biology course my freshman year, and still all through that, absolutely hated it. But I think somewhere in the space of learning about brain imaging and becoming very interested in that, I started to see biology and the rest of science through that lens. And a little bit of this was kind of falling for this, this reductionist trap of, all your thoughts and such are really just like these cells bouncing around doing their thing. And so, at least that at the time gave me a way to kind of appreciate biology a lot more. And by virtue of that, all the other science courses that you had to take to study biology and get a degree in it. But I was so fascinated by it. And so I ended up doing that and taking more and more neurobiology classes and such. And in the process of all that, I think I also just really developed an interest for the first time in learning all that was out there. And, again, it was being able to see things in context, right? Now, all of a sudden, all this stuff was really interesting. And it was all like different pieces of a big puzzle. And so, I got very interested. I'll focus more on the education side here. Obviously, research and research experiences were a big part of this, but, I became more and more interested in brain imaging. And that brought me to, for grad school, Washington University in St. Louis, which was a big hub for this sort of thing. And there, I met some fantastic people who really inspired me even further to broaden my interests and think about questions more broadly and in a multidisciplinary way. And I think I really enjoyed my time there and became more and more interested in neuroscience and how all these different scientific disciplines and the tools from different disciplines and such, how all of them kind of have, it seems like, something to offer in terms of helping us understand ourselves better and understand brains. And so, yeah, I would say grad school was a enjoyable experience for me in St. Louis for further pursuing this work. And then I think my research, ultimately, I naturally found myself gravitating more and more towards the computational side and kind of systems level perspective. And so when it came time to choose a postdoctoral direction, I think I was really lucky, one, to land up in the position that I'm in now, and this fellowship opportunity that came along in the Seattle community, which was kind of on my radar as a real hub for computational neuroscience, and really just in general, especially the people I'm working with now, real experts in machine learning and dynamical systems and such. And this was a place where I could see some of these ideas, I think, really coming together in a way that I think haven't fully, realized their potential yet in neuroscience. And so I thought being embedded in that kind of community and then also having the opportunity to have a fellowship that would allow me to learn those skills and also start to try my hand at applying them in neuroscience. I think that was a really cool opportunity and really beneficial to my progress. And so I guess that's a long-winded way of retracing my academic trajectory through those three institutions.
Yeah, I think your package did really nicely. We were exactly hoping for that kind of from point A to how you got here. That was awesome. So it looks like you had that natural curiosity about yourself, as you mentioned, the human psychology. What are thoughts? What are free will? But either you could tackle it just from the psychological perspective, but then you're like, let me see it from the biological perspective and learn more about that. Is that right?
Yeah, I think, and I want to pay respect also to the people who have thought about these things deeply and in a very meaningful way in psychology. But I think my experience early on, and actually I'll say also, you said it started from an interest in myself. Honestly, I would say it started more from an interest in other people.
Oh, okay.
So I was actually very interested in understanding, other people and their thought patterns as well. And so I think what pushed me from psychology to more neuroscience proper would be, I think I was starting to take cognitive psychology courses, and it was supplemented by cognitive neuroscience. And I think cognitive psychology, I had the experience that I think a lot of people have shared in trying to get into this space, which is that there are a lot of different competing theories, a lot of different models, and it's hard to precisely specify these in a language that you can compare across different frameworks and say these predictions are more accurate and better support this model versus this model, right? So there's a language barrier, certainly, that prevents, a lot of theoretical progress in the field. And I think supplementing that with cognitive neuroscience and being able to point to particular mappings to biology and such, I felt like it was just opening up. It felt like a fun time to be getting into that space where it was just opening up a world of new possibilities, where now you're able to address some of these psychological questions and such with the advantage of also having some additional new context to go off of. So I think that opportunity really pushed me down that path. It felt fresher.
Yeah. For me, it was the other way around. I used to be like, okay, physics, electron, neutron, and then zooming out chemistry and then zooming out biology. So I think input, output, that's it. And then very reductionist approach. And then later in my sophomore year or something, then I started to be interested in general psychology or like social psychology. So
cool.
That's why I asked that question.
Interesting. Interesting.
For me, it was absolutely reversed. That's why I found that.
Well, you and like all the Nobel laureates and such, right? So you have good company in terms of kind of, developing these interests in high level psychological topics and such after you've kind of done your diligence in physics and such. So
Yeah,
Interesting.
I guess my background is also more of like engineering based, and then I
Cool.
have come into this neuroscience world where I have to rephrase how I'm thinking. But I'm
Yeah.
wondering, since you came from the opposite world, like how did you make this transition into manipulating data, just doing computational work? Since you mentioned that you did not like science at all to begin with, how did this transition happen, and how are you now thinking about all of these questions that you were asking yourself previously?
Yeah. Good question. I almost hesitate to say transition because I don't know if I, would fully call myself any one of these things, but I think, so I guess it is true that more typically people go the other route, and I think in some sense it can be harder to later develop kind of it's almost like a higher barrier, right? To develop the kind of the quantitative foundations and such to be able to kind of post hoc go in that direction and bring engineering and machine learning and such these tools to bear on your biological questions. But I think what motivated me to go that route, and I think for all the same reasons, I think because you don't see that path as often. I think what ends up happening is that a lot of the work in quantitative neuroscience and such, it's not always kind of fully motivated from the biology side, if that makes sense. There are kind of tools that are being developed in the quantitative sciences, and oftentimes you want to test these tools. You want to look for a biological question that might be answerable from this framework, right? And I think this has led to a lot of progress, obviously, in quantitative neuroscience. But I think it left open some opportunity too, to if you really start from biological questions and say, okay, what if we need to just go out there and learn the right tools and maybe develop the right tools in order to address those questions computationally. And so yeah, I think that's what kind of inspired me to branch out in that direction. And of course, it's as challenging as anyone else would find, kind of moving in a different space and such. But I think that's also been a lot of the fun of it for me. I think the most fun aspect of research in general, academia, has been for me just being able to continue growing and learning. So
Yeah, that's awesome. The place you're working right now is, I suppose, is very multidisciplinary, right? Allen Institute. So there might be some scientists coming from engineering background, and then some from maybe even electrical or computer scientists. They come together. You all look at the project together, and some go from, okay, I have these tools, let me look at the biological problem, and then you come from another direction. I have biological problem. Now, what tools would solve that? So I think that's what you were trying to address in the beginning when you said a lot of computational people coming together in Allen Institute. Are you finding that in University of Washington and Allen Institute? Has that been your experience?
yeah, I think it might be comparable even to, I think you guys all have a really, really great and really rare interdisciplinary community here, right? Which I'm very much looking forward to experiencing more of. But I think it's a very similar thing where you have expertise represented across a range of disciplines that are all relevant to these questions that we're all after. I think in Seattle, the real unique strength is this there's a big neuro AI community. There's right, even from industry, there's a lot of expertise represented in kind of modern machine learning methods and such, deep learning. And so there's nice infrastructure that I was able to piggyback off of in terms of already existing collaborations or spaces for conversations that involve neuroscientists as well as, pure computer scientists and everything in between. So I think what's worked out well for me, what's been a real, lucky opportunity has been this fellowship where having the flexibility and independence to kind of, the integration is happening, but not necessarily always right around these exact question that you care the most about, right? And so having the opportunity to at least be the person to kind of go around to these different communities, right, and learn from their expertise and try my best to help bring that together. I think that's been the really cool opportunity I've had in Seattle.
Okay, so I think this is a great segment to segue into the research you're currently working on, or what have you been working on during your fellowship? Can you explain a little bit more about just what questions are you interested in and how are you approaching these questions?
Yeah, absolutely. It's kind of funny to think about, and I imagine it's not such a unique experience to me, but I came in with a particular, I had a certain background for my PhD, and I planned to kind of branch out a bit more during my postdoc, and ended up asking questions that weren't as far removed as I might have anticipated. And I think maybe that's a common experience. I talked a little bit about the kind of career-related motivations, right, in terms of wanting to gain more computational experience and broaden that skill set. Scientifically, I think, nearing the end of my PhD, I had come across some interesting questions related to brain state dynamics and arousal. And I think the theme there was covariation everywhere, right? And I feel like I didn't know how to think about that. I felt like I knew enough to suspect that a lot of the ways we approach this question kind of really restrictive assumptions and just weren't necessarily respecting the intrinsic beauty of the problem. I'll say it in that way. The kind of covariation we see is really a signature of just this remarkable level of coordination throughout the system, like totally unparalleled by anything that we can create. And I think that's something that's new for engineering sciences, for that reason that we can't create something like this, and for a lot of things like time series analysis and such, and even standard practice in biology and experimental methods. And so I felt like I didn't fully understand how to deal with that. And I think coming to Seattle, I was eager to start on different questions as well. And, this problem of arousal, which I was working on during my PhD, also then kind of inadvertently, but it didn't seem to go away, So it's almost like, okay, if I think of now, I'm thinking of an entirely different recording modality, let's say, and I'm focusing on this specific brain region, but I'm still, at the end of the day, studying an awake animal, and this awake animal is not doing the task that you necessarily want it to do because it doesn't fully have that kind of, maybe a better way to say it is it doesn't have the motivation, but there's some kind of intrinsic dynamics to the organism that play a role in what it's doing. And I wasn't able to fully leave behind the concepts of brain state and arousal and organismal state and such for that reason, that you can't, once you kind of start to appreciate it, I feel like you kind of realize, or I kind of realized and kind of felt like it was there, whether I wanted to pretend it was or not. And so that's what kind of pushed me to continue working on these problems related to brain state dynamics and arousal, was that, okay, if I ever want to go to any other aspect of neurophysiology and such, I still kind of need a way to do justice to the fact that the organism is effectively a continuously evolving context. And I don't want to take my recordings and such out of that context. I want to be able to understand that context so that I can relate my findings back to all these other aspects of the organism. So yeah, that's how so I've kind of stuck with this theme of brain state dynamics and such, and approaching it, again, a big theme for me has been, okay, how do we get serious about studying this quantitatively, and do we need to potentially develop alternative frameworks for thinking about this and for measuring it?
You mentioned moving on to explore electrophysiology and so on. Can you tell us a little bit about your current methods and techniques that you do to record?
Yeah, well, to be clear, I'm totally on the computational side. So I benefit immensely from very talented experimentalists, and this has been the case even through St. Louis. I was very lucky to work with highly trained experimentalists who knew how to take these ideas and questions and pose them in the right way, in the case of hypothesis-driven research. But then more generally, I've used a lot of open access data, that very hard-won open access data from experimentalists looking at all different types of modalities. And I think it's important to recognize that, because that has, that's the only way that I've been able to look at different modalities, right? I think the reason why things in neuroscience, like different subfields, essentially you could organize them based on experimental modalities and such. Part of it is just because each modality requires its own level of expertise to be able to deploy in a very rigorous manner and a very careful manner. And so it requires a certain degree of specialization. And, where I've kind of tried to complement that is by saying, okay, I'm useless in actually collecting experimental data, but maybe through computational frameworks, I can play a role in helping to bridge these communities by working with the data that they've collected and putting it into kind of common spaces.
Thank you. Yeah, now that makes sense. I heard not a lot of people know how to do PATS-CLIM correctly. Not every institution might have fMRI access and everything. So I see what you mean. Like every experimental modality, you need experts in that domain to collect the data, and then having access to that data and bridging those different modalities together. So what modalities are you currently working with? Like what kind of experimental data, if you want to share that?
Yeah, sure. At this point, I've worked with a good range. I think some of the work that we recently put out a manuscript on, and that I'll be, I think, talking a lot about with people here in Boston, is wide field calcium imaging. And this is a nice tool that bridges large-scale spatiotemporal brain dynamics with the accessibility of the mouse model as a genetically, being able to do all the different manipulations we could do genetically with this model opens up a lot of avenues. So wide field calcium imaging has been one of interest for me in recent years. And moving forward, I think I'm also, I'm very excited about the tools that we have now for doing high density measurements at cellular resolution. And so I think this just provides another window and a new type of information for me in particular, I should say, to actually study and respect the diversity of cell types, for example, but at the same time, coming at it from this, in my case at least, the systems perspective and saying, okay, we know something about these organizing principles at the system scale. How can we use that information to contextualize these cellular level phenomena? And at the same time, can we learn something about how these different cells, basically, how do they see their role in the big picture, so to speak, right? So I think that's a direction that I'm really excited about in particular, is these kinds of high density cellular level recordings in mice.
So what kind of recording are we talking about? Is it high density electrophysiology?
Yeah, sorry, I meant to mention I think the two kinds in particular, although obviously, and especially at a place like BU, you have such an extraordinary diversity and are at the forefront of developing new tools, right? But the ones I had in mind that have kind of really taken off already are 2-photon calcium imaging and these neuropixels recordings, right? So these silicon probes to do high density electrophysiology across the brain. I think these are really exciting tools.
Yep.
Those sounds, awesome plans. Can you tell us more about some of the most exciting research findings that you have so far? What were you surprised to see? What were you expecting to see? Or in general?
Yeah, okay. Good question. Let's see here. I think maybe the biggest theme, if I were to take, there's almost like a roughly continuous thread from some of the stuff I was working on early on in grad school to what I'm doing now that is accompanied by some interesting surprises and changes of perspective and such. I think where this understanding sits right now and this focus on arousal and such, that all I think is important I think it's kind of interesting to think about how that came out of the data itself. I never set out to study arousal. That was something that I tried my best to get rid of and I couldn't. And so I accepted that maybe it's here to stay. So, the story behind that and these, in 2021, I think, we had a paper on these traveling waves throughout the brain, and a couple other groups, Max Schein's group, Shao Kielholz's group, Xiao Wu, they've done some beautiful work also characterizing these large-scale traveling waves. From my perspective, how we arrived at that through the themes we were working on at Washington University with, and this is in Mark Rakel's group, for a few years now, by that time, Anish Mitra had been carrying out some really, really interesting analyses revealing these interregional delays between brain regions. And I think we were gradually recognizing that global phenomena seem to play a big role in that. And as you may know, global phenomena, really in any modality, but especially fMRI, have always been a source of contention. And I think, this was also at a time when there was a lot more controversy about, things have changed a lot, I think, in recent years with large-scale electrophysiology and, again, these high-density recordings and being able to study awake animals. And now you talk about arousal and you talk about brain-wide covariation, and it's like, yeah, we see that everywhere. But at the time, I think in a lot of ways, neuroimaging kind of was ahead of its time in that sense. And so you saw these things also, obviously, from the get-go in neuroimaging, but you just kind of had to assume it was artifact, right? There was no precedent for thinking that there was this covariation everywhere. And so I think, with where this temporal delay research was going, eventually, like I said, we were realizing that there was this big kind of global component of it. And around the same time, I guess pushing on this theme, I realized that it was basically this beautiful kind of brain-wide traveling wave structure. Like there were these actually, at that time, we had just been studying it in terms of pairwise delays, and it wasn't clearly mapping onto spatial gradients in the brain. But at a certain point, especially when looking at it from the global perspective, that's when we started to realize that there were these clear gradients. And so that implies a traveling wave structure. And this was also around the same time, a bit after Margulies et al. Had their really, really great insights about this kind of continuous gradient-like aspect of functional organization across the brain. And that also fit neatly into this picture, so much so that it became clear or highly probable that this traveling wave is kind of responsible for introducing these slight differences in the signals that you measure across brain regions that manifested as gradual variation in so-called functional connectivity or correlation structure. Anyway, where I'm going with all this is that I thought these traveling waves are cool, and there was this beautiful organization coming out of that, and I was trying my best to get a way to study this in a totally data-driven way to pull out these waves. And I should say, I guess the technical term I'm kind of alluding to here is in an unsupervised manner, right? How can I just extract out these traveling waves? And I couldn't for the life of me. I could use some off-the-shelf methods and such, and it would work, but it wasn't really capturing the phenomena in full. Like it was kind of doing some damage to the natural manifestation of the phenomena. And so the best I could do, to my own frustration, was to time lock it to external indices of what we would call arousal. So things like heart rate, respiratory rate. And I didn't want to do that because that invariably, one, I didn't know how to think about that. I was like, I want the story to be about these really cool traveling waves. Oh, by the way, they're coupled to arousal fluctuations. What does that mean? And then second, I didn't want to do that because that opened up this line of questioning about, oh, okay, so do you mean it's all artifact? And I didn't want to engage with that. And so I tried for the longest time to study it in a way, in an unsupervised manner, to get rid of the arousal indices. And I couldn't. It just seemed so like the cleanest way to get them out was to time lock these fMRI signal fluctuations to an external measurement of arousal fluctuations. And so that's what we did. And, a couple experimentalist shout outs here in order. One of them first of all this data that we ended up using was publicly available to the Human Connectome Project, which was just a incredible effort and has benefited the community immensely. But Rafi Malik and Dov Yellen, they were from the Weizmann Institute in Israel. Early on in my PhD, when I started going down this route, they provided their previously collected simultaneous pupillometry fMRI data. And that played a big role. It didn't end up playing a big role in the paper we ended up publishing, but that played an enormous role in my thinking at the time, because that was the first arousal index that I used and found that was the cleanest way to bring out these traveling waves. Like it just fell right out of the data, and anything else I was doing at the time didn't work. So that was the reason I stuck with it. And then finally, what allowed me to maybe, sleep at night in grad school eventually was the hero, now Takafuji, who, again, you have to revisit the backstory to like the context of, even today still, but at the time in particular, you're talking about fMRI and you're talking about arousal, and you're definitely talking about an artifact. Like it's, these were major concerns, and there was already a line of research showing that, systemic vascular fluctuations correlated with global fMRI activity and such. And so there was this really cool story emerging, and it felt like it had a lot of implications, but I could never really, at the time, I was reasonably comfortable saying, okay, it's an important phenomenon and we need to, it deserves study regardless, but I can't prove at this point, right, that this is, so-called neural and not hemodynamic. But during that stretch, I guess that dealing with that frustration and that uncertainty, when I look back on it, I think that played a big role in shaping my views even to this day, because eventually we reached out to now Takafuji at the time, at least, Riken Institute in Japan. And he shared this one of a kind. I mean, it was already publicly available, but because he was just way ahead of his time in terms of data sharing and such. But they had this incredible hemisphere-wide electrocorticography data set. And that was the data we needed. I mean, EEG didn't have the spatial resolution to talk about traveling waves of this kind and know for sure that it is a traveling wave. And ECOG usually is performed on just a relatively small section of the cortex. This was one-of-a-kind data to test this hypothesis that these traveling waves we're measuring, even though they're linked to these ongoing arousal fluctuations, they are also relevant to electrophysiological activity.
And I suppose those were in human subjects?
That was in awake macaque monkeys.
I see. Okay.
Yes.
Yeah.
And so this data is publicly available. They did an amazing job of curating the data. And it was, it took like a half hour to download the data and confirm the findings. And I still remember that day. Like, it was just amazing. But so it worked out. And long story short, now thinking gets into my more recent work, like it's all kind of coming to grips with this idea that these traveling waves, but it seems to reflect the same process governing these arousal fluctuations, et cetera, and everything's co-varying with them. How do you study that? But going back to this point that the uncertainty influenced my thinking, by the time that we were able to confirm the results in electrophysiology, I had long left behind the view that just because you see something in, for example, peripheral physiology and vascular activity in the hemodynamics, whatever, that doesn't mean it's an artifact per se or anything like that. Like, I started to kind of push back on these kinds of views just in my own head and realizing how much of our language and a priori assumptions shape how we're treating the data. I mean, those are, I don't know, I've never seen a brain that doesn't work with blood flow before, right? Like, you kind of need hemodynamics, you need a metabolism. These are arguably more fundamental than neural activity in certain respects, right? So by that time, I had already kind of grown very comfortable with some of these ideas that are ways of thinking about one-to-one causality and the idea that you could regress out one signal and have that meaningfully remove whatever was generating it from the data. The idea that you could meaningfully tease apart, you could talk about neural activity and non-neuronal activity. By that time, I had already come to appreciate that these are all gross oversimplifications. And so when the electrophysiological data became available and when we were able to confirm the result in electrophysiology, it wasn't, oh, we validated that this is actually a neuronal phenomenon and therefore we should care about it. It was more like, okay, for a lot of people, yes, it's that validation. But here, it's also like, wow, this also does, in fact, entrain the electrophysiological activity as well. So all the more reason why it appears to be important. So I would say that's again, another very long-winded answer. But, going back to your question, I think that's probably the most interesting and spontaneous thing that's happened over this research trajectory is how I ended up in this space of arousal and such. Oh, and then I guess the final piece of that story is at a certain point, I realized I had no idea what I meant by arousal. And then I also realized that most people also did not know what they meant by arousal. So I felt, some company with them, but at the same time, I was like, all right, we got to get serious about this. So that's how I started studying arousal. Never set out to actually study that.
Where do you think your research in this field is moving on for the future? Where do you see it just progressing?
My research or this field being...
Just like arousal, exploring the brain, exploring arousal states.
I mean, I think there's a lot. I find myself more hesitant to speak of where the field is heading because I don't know if I have the best pulse on it. But I think in general, I would say it would probably be relatively non-controversial to say that the field is increasingly, even already, but increasingly so, talking about brain states, recognizing that we're studying a dynamic organism, a dynamic brain, and everything that we measure is, in some sense, state-dependent, it seems. And, we got to start unpacking what we mean by brain state and the things that compose it, and how do we make those more amenable to scientific research and quantitative research. So I think this general theme of brain states is really taking off. And I think that's kind of a community effort to really better understand these brain states, both in health and disease. I think disease is a bit further off in terms of we just don't even have a good basic science understanding of what comprises brain state at the moment. And I think for my research, yeah, it's kind of centered on that as well. And continuing to make progress on specifically, I'm pretty tethered at this point to a dynamical view of brains. And so for me, brain state is directly analogous to when you talk about the state of a dynamical system. So this is a dynamical system that has a lot of things going on a lot of different spatiotemporal scales, right? And so for me, brain state is a question of better understanding this thing that we're construing as a dynamical system. So can we identify different time scales of dynamics, how they play out spatially across the brain? And we have to kind of use everything in our arsenal, different experimental modalities, et cetera. And on the flip side, just as we're using those modalities to better understand brain states, in turn, I think an evolving understanding of the brain's dynamical organization is going to pay dividends in terms of understanding the measurements that we're collecting from these different modalities.
Thank you. That's awesome. I think now we want to segue into more like question and answer type of thing. You packaged all of your academic journey and then your interest, how you got here, and the research question, arousal, everything. Now I think our question and answer session, we would want to...
Rapid fire.
Okay.
Yeah.
Question and
Cool.
answer.
Free association.
Yes. So for first question we have for you is, what advice would you give young aspiring neuroscientists or PhD students that are in the field very early for, let's say, first year, second year, or prospective PhD students?
Yeah. Oh, okay. Kind of a non-answer, but I don't like giving advice. And I think my advice would be to always remember that any advice you get is from people who have different goals than you. And so I guess if I had to push that into a concrete form of advice, it would be your goals are going to evolve over time, but always be clear and try to push yourself to identify what things are most important to you as a human being, not just as a scientist, right? And keep those goals in mind and use that as a filter whenever you're getting advice from people, because you're going to get a lot of conflicting advice from different sources. And there's a way to get use out of a lot of advice, but I think you really need to stick to your principles.
That's really powerful. Yeah, not the answer I was expecting, but that's really powerful. Yeah, often academics, PhD students, I guess, or industry in general in career, we sometimes tend to tether our identity to one project or one thing, and then every advice is looked from that filter. So that's
refreshing
really
to see your perspective. Yeah.
Thank you.
Yeah. Okay. And next, very quick question. Well, Sudan and I are engineers, and we often think from an engineering perspective. So we're wondering if you could go back in time, like what engineering tools you wish you had during your studies.
Like experimental tools?
Whichever you prefer, software, experimental. Like what was something you were lacking?
Well, I don't know. I wouldn't say I have too many regrets about how I approach things and such, but I think if I would have changed anything, maybe I would have not that you'd call it an engineering tool, but I think at least as a quantitative tool, I would have kind of adopted a dynamical systems framework and formalism earlier on and gotten into modeling as a tool to ground everything I was doing. I would have started that practice earlier.
Okay. We'll take the computational tool as engineering tool.
You'll claim that.
Not to offend any computer scientists, but we'll take it. Are there any tools you wish is, like right now, you could see it in the future, maybe not flushed out or developed, but is there any tool or computational thing that you see it's coming, but like you wish you had it right now to make your research go faster?
Oh, okay. I think I'm maybe understanding where you're getting at a bit better. Okay, I think the future. I think I'm really excited about closed loop experimental designs, and engineers have been indispensable for pushing the frontier of like brain-computer interface type designs, neurofeedback, et cetera. And I think these tools, I mean, we're only just starting, I think, to really capitalize on not only obviously we've known for a while their benefits for clinical translational neuroscience, and that's been a success story in a lot of ways. And I think we're just uncovering how useful they can be even for basic systems neuroscience and even in species like rodents and such. So closing that loop between brain activity, behavior, and environment and such gives it a really unique platform for studying these things.
If you had unlimited access to the data that contained the entire brain map, either mice or humans, what sort of research questions would you be interested in asking?
Maybe it's a hot take. I don't think my questions would change very much. I think already all the data sets we have in front of us are higher dimensional than my brain can work in. And so I think still an ongoing effort to understand how to approach high dimensional data and such, but I'm not lacking for data. And so I think if I had the whole map available to me, first thing most people would do, I think, is, if we're being honest with ourselves, is some type of dimensionality reduction into our understanding.
Yeah.
So, yeah.
Fair. Well, we thank you for coming onto our podcast. You will going to be here at Boston giving seminars, workshops. So we thank you for just joining us. And yeah, Sudan, any closing remarks?
Yeah, thank you very much. We got a lot of interesting answers from you, the ones we did not hope. So we had to change our thinking while we were asking you the follow-up questions, which was really interesting and amazing. So to wrap up, we have a small token of appreciation for you from BU Neurophotonics Center. And our producer, Sung, is going to hand you over.
Oh, that looks nice. Oh, thank you. Thank you so much. This will come in handy because I definitely underdressed for the... It was kind of windy this morning. No, before we go, I just want to say thank you so much for inviting me to come on your podcast here. And I think it's really cool what you're doing here. And I'm very grateful for the opportunity to even just be visiting the research community and have opportunities to engage with the students, with the faculty here. This was a lot of fun for me, and I'm glad I succeeded in keeping you on your feet. That was fully the intent. No, this is great. Thank you so much. I appreciate it.
It's a pleasure. Thank you very much.
Yeah, thank you.
Thank you for listening. If you liked this episode and want more like it, please take a moment to share it with anyone you think would enjoy it too. Next time, we will be joined by Dr. Emily Stephen, an assistant professor in statistical neuroscience here at Boston University, as she discusses how she is overcoming gaps across spatial scales and shares her role in the data science aspect of the Neurophotonics training program. This episode was made possible by the support of Boston University's Neurophotonics Center and listeners like you. Thank you.